Good morning, everybody. This is Rojing Wang. We are in Seed Science and Technology section of Saskatoon Laboratory of CFIA, Canadian Food Inspection Agency. Today, Jennifer and myself, we're going to talk about this referee. The reason I want to stop Jennifer is I want to give a little bit of background about this referee. So every year we try to provide a virtual identification referee. The purpose is slightly different from other referee. If we provide referee for germination or other type, it's mainly for method training, gap identification or validation purpose. And this virtual ID mainly is for training purpose. So for this training purpose, in the past, we provide virtual ID for weed seed order species. And from now on, we will focus on training on different families. So this year, we chosen 2 families. So we can, with a combination of training different level, so this level is toward people have a higher identification level to distinguish the similar species. Then the referee have 2 parts. The first part is exposure for you. So you do ID without training, this intensive training. So that kind of message, I heard many feedback, people start worry about their performance. So you don't need to worry, this is just training. So the first one is exposure. The second one is today is Jennifer to provide. So this is a new format for you. And if you have any feedback, please do send us by email communication. So next year we can better prepare for your need. So I repeat once again, this is a training exercise. It's not a PT, not a performance purpose. So now back to Jennifer.
Oh, thank you. Thank you for that. Very good. And please feel free to come in as well. I will have some slides that will cover the participation and some of the recognizing or how many labs recognized those species. So we'll cover that. We'll have a couple of slides there, and at any time, Rojing, if there's anything you see, please come in. All right. So I just put up the objective screen here while Rojing was talking, and I should say at the top objectives of this referee webinar. And in this particular webinar, I'd like to review my method of identification of, as you folks no doubt have seen, all the similar species from this referee, which includes some with regulation significance and others that are look-alike species, and some that are found, you may not have seen them before, and some that you may have as common impurities. And there are also those that are found in Canada and the United States. So I'm also going to show you my tips and tricks, or what I do when I approach these very similar seeds, to be able to separate those species in these 2 genera, the Linaria and the Solanum, and hopefully using the training provided in this webinar may shed some light on some of the questions. All right, and I should have put this up while Rojing was talking. So this is just a little screen. You may see it again with a mail out specifically about the referee, but I just thought I'd put this up here, that we had a total of 40 analysts that participated in this virtual seed ID. Some of the feedback already is that, as Rojing addressed at the beginning, these were difficult IDs. Many of the species you folks may have never seen on your slab before. Another suggestion was to do multiple choice questions and perhaps have a webinar such as this first. But of course the images, and I pass this along to our staff here at SSTS with the NSH that have taken some excellent quality images over the years, so that's excellent. And as Rojing said, definitely communicate by email if you do have some more suggestions or any feedback on this particular webinar, and you see our email there at the bottom. And this webinar will be recorded and available, so if many of the slides move a bit quickly. We only have about an hour, so you can definitely go back and review on the recording when it is available. Excellent. So here is the species in the referee. In the referee, we had 9 Linaria species, so species that were only found in the genus of the Toadflaxes of the Linaria. And then we also had 10 Solanum species, only that in the genus Solanum, and 19 species in total, so there's quite a number of species. And you may be able to recognize some of these names. And some of these names were definitely new to me, so this was a learning experience for me as well. So I do have a slide here of some terms that I will be using in this webinar, and I won't spend too much time on this slide. This will be available in the recording. But I'd like to draw your attention to the 2 terms at the top, because I definitely will be saying these over and over again as subtle, very specific seed features. The first is what I call reticulation. And this is a technical term that can mean ridges, I should add grooves, as well as veins and nerves on the surface of a seed that forms a net or a grid pattern. And it may be irregular, it may be rectangular, it may be square. It can be composed of either ridges or grooves. So that's the actual ridges themselves. In between are the interspaces. And these are the space or cell formed from the intersection of these lines or grooves of the reticulation pattern that I mentioned above. Both the reticulation and the interspaces will come important in many of, to distinguish many of these similar species. So for the 2 genera, I found that there were specific places that I looked on the seed to find those features that distinguished between similar-looking species. And in the past, I've called these information points or concentrated areas of seed features. Those are the places that I found where the differences lie in these genera. So seed features, those structures or qualities of a seed or fruit that are observable, describable, and hold information useful for identification. And I found 3 different areas on both Linaria species and Solanum species seeds that can be studied, those information points, to distinguish between similar species. So for your Toadflax seeds, I found that the entire shape of the seed held significance, the surface qualities, which I'll detail, of course, in the next few slides, as well as the edges were important holders of information for me. For the Solanum seeds, I found, of course, the size. I've got it right there, diameter, I mean. The surface, and you can see already those ridged reticulations on both of these flat faces. I'll be talking about those, as well as the surface qualities near the hilum. And in many Solanum seeds, many seeds in the Solanum genus, the hilum area, though along the narrow edge, often is pulled out, the seed tissue. So where the hilum is on these seeds is often quite obvious from the side. Excellent. All right. So the way that I'm going to break down the exercise that I use to distinguish between the species in these 2 genera is I used rather I aggregated and then kind of divided from the aggregation rather than starting with the seed itself and then try to feed it back into the genus or the other seeds of the genus to try and find differences. I find that that's a very labor-intensive way of distinguishing between similar species. It's nice to collect all the similar species in a large group and locate one strong morphological feature that could divide all of the similar species into 2 broad species groups, fairly balanced. These features are usually quite obvious. Then taking those 2 groups and finding a feature that could divide each group further until individual species or extremely similar species groups remain at the end of the exercise. So consider them all and then start finding very broad features that could break out, that could break the species apart into groups, and then find other features. And those features become a little more subtle and detailed, where you would have to see them under the microscope, while the initial features, those broad features, can often be seen with very low magnification. So let's go right into the Linarias, the Toadflaxes. So here's a slide that shows the overall correct ID or what species were correctly identified. There were 2 species that were identified over 90%, and both of these do occur on the Canadian weed seeds order. And definitely one of them, the yellow toadflax, is a common impurity and is easily recognizable. You'll notice that all of the ones, there's 6 species over 60% recognition, and a lot of these are either on the Canadian weed seeds order or have been, well, pardon me, because they're on the weed seeds order, they're often trained. So you have had, analysts have had experience in seeing pictures of these seeds and maybe having other webinars or having them talked about, and you'll see that that training is paying off. There are some species down here lower than 50%, and those are species that people, including myself, may not have seen before as an impurity, and this is more for exposure. And if the rest of the high level of recognition is of any indication, training and exposure to these seeds will also increase the recognition. So really, I think this was successful in showing that, as well as exposing you folks to how many species can be found in the United States and Canada. Now notice up at the top, I have a synonym. What I did when I took a look at the data is I made sure that any of the answers given, even though they didn't match, of course, these names on the bottom, they could be old names or synonyms. So I made sure to take a look at those and to give credit when a synonym was used, because you folks did recognize it. And the synonym that was used for Linaria dalmatica, Dalmatian toadflax, was its old name when it was a subspecies of Genistifolia, and I'll cover that in a separate slide. So that was one of the synonyms that were answered, but it is considered correct. So the Linaria seeds, just to show you what the fruit looks like, folks may never have seen the fruit on their slab, and you'll see that it is a dry capsule, and you can see the valves on top when the capsule is mature that releases these seeds. Now you'll see that this likely is dalmatica, Dalmatian toadflax, and you'll see the very polygonal or angular shape of them, and that's because they're kind of crammed into the capsule, and so you'll have irregular shapes. You'll often see these in capsule fruits. The seeds from these fruits tend to be irregular because they're kind of just, it's like a free-for-all in there in terms of maturity. Some are small and some mature large, the ovules, that is. So here we have up in the corner are some blonde seeds of Linaria dalmatica. And why these seeds look different is because they're immature. Many of these dark-colored seeds will be blonde when they're immature. It seems that they darken up as the last leg of maturity before they are shed. So if the fruit breaks apart or there's some left on the bottom that have a delayed maturity, you may see these in your samples, and they are still considered seeds and Linaria dalmatica. So taking a look, here I have a decision tree is what I used, or how I kind of tabulated my identification, how I went around the identification of this big group of similar-looking Linaria species seeds. So as I said, I looked for that one feature that could split apart the big group, the 9 species, into kind of a balanced group, or pardon me, 2 groups of about the same number of species. And I found that quite easy in Linaria with the seed shape. There were 2 types of shapes, or 2 categories of shapes in the Linarias that we saw. Some that were round for species with a marginal wing around a wing around the outside of the seed, looks like a fried egg. And then the other type are the angulars, like I showed you with that slide of Linaria dalmatica, in that they are seeds that have many faces and many angles and don't lay flat on the slab. They come up a little bit. So in the round seeds, what I took a look in my round group was what would be the next feature that could split my round group into a number of species groups. And I found that the texture of the seed center is what popped out to me. And I found within our group of 9 species that were 3 different kinds of textures. One species in particular was ridged, so I was able to carve that off right away. That's 3 birds flying. Then I took a look at those that were smooth, I took a look at those that were tuberculate, and that's just a technical term which means warts on the surface. After that, I took a look at the shape of the seed center in order to split my smooth seeds and my tuberculate seeds apart, and I was able to get down to species just with 3 steps. So notice when you do get down to a species, in order to characterize that species, you have at least 3 features that are characteristic of that species that you could enter into a table or into a program that allow folks to recognize the seed. Now, I'll detail the seeds and pair them off and show you pictures, so I'm not going to spend as much time on this decision tree. And I'll have these for the angular group as well as the solanums. So here's a pictorial representation of my first split, which was based on seed shape. And you can see the fried eggs over on one side, the round shapes, and then the polygonal, irregular, angular shapes, and they split apart fairly evenly. Taking a look at the round seeds, I told you that I looked at the center to see whether they were tuberculate or if they were smooth or with ridges, and again, it split it apart fairly evenly. I was able to carve off 3 birds flying, Linaria triornithophora, by these ridges. So they're kind of warty, but they form lines. And they do look different in that the marginal wing is quite reduced. And it's a thicker seed in the middle as well. It's kind of a robust seed. It's hard to see that in hand. Pardon me, when you don't have it in hand. But that one came off. And there's 33% of you folks that did recognize it, so that was great. Now, looking at the one with the center of the seed smooth, I had 2 species with a smooth center. Linaria, well, 2 of them. Let me go on to the next slide. There we go. So I've collected them all onto this slide, those 3 species that were left that had a warty or a smooth surface. So in the warty surface, I had 2 species. One was the yellow toadflax. The vast majority of you folks got it correct. Fried egg look, warts right in the middle, or tubercles, and also I found that prostrate toadflax also can have warts. Prostrate toadflax can also have a smooth surface. These are the 2 bookends of the seed texture, because there are also seeds that have only a few tubercles and that are mostly smooth. So there's a continuum between these within the species. Now, prostrate toadflax is a flower seed, so I'm not sure why some are tuberculate and some are smooth. However, always, I have noticed that prostrate toadflax seeds have a half moon or C-shaped center. So remember, I got it down to texture first, then I looked at the tuberculate species and found that the shape of the center was different. Then I looked at the smooth seeds, and you'll notice, or when you go back, you'll notice that I had 2 species, and that prostrate toadflax overlapped with the smooth, in the smooth category as well, along with the main one, which is melancholy toadflax. Now, melancholy toadflax is indeed always smooth. However, the center, which is normally that irregular kind of fried egg, roundish oval shape, some can be the C shape. It can contract into the C shape. So unfortunately, at the end of this one, if I can go back, there was some overlap with the seed center shape, unfortunately, which is what I saw. All right. Now going on to the angular species. So again, we had our round decision tree we saw earlier. Now we're going to look at the 5 angular species. There was one species that was quite distinctive looking at the seed texture. One of them was ribbed. That's your Moroccan toadflax. And then the other 4 species had that reticulate, that network of ridges on the flat faces. Now we're starting to get into very similar looking species, so what I tried to do is look at the surface quality. I noticed that 2 of the species in the referee were had a dull matte surface that didn't reflect light back, while 2 of them had a shining surface, a metallic wash to the surface, or oil slick is another way I've heard it described, and I split those into a group of 2. Within the dull, I took a look at the edges. Now we're getting into super subtle. And in the metallic, I looked at the interspaces. So let me show you in the next few slides. So like I said at the beginning, when I looked at the angular seeds, I found one that was distinctive with a ribbed surface rather than a reticulate surface, and that was Moroccan toadflax. And of course, it is recognizable, and 80% of you did recognize it. Though for myself, I will say that I've never had it come in a sample as an impurity as of yet. And the reticulate surface, of course, there were the 4 species. Now, as I said, I took a look at the surface, starting to find what features can split this group of 4 apart, and I did find that the surface quality was noticeable in that you had either a dull or a shining surface. So taking a look at the dull surfaced ones, now you see they're starting to look quite similar. The striped toadflax, which is on the Canadian weed seeds order, is a larger seed as compared to his lookalike, the purple toadflax. And indeed, you can see the disparity in the recognition, likely based upon the training that's given to weed seeds order, or the resources that are given for training in the weed seeds order seeds. So the striped toadflax, take a look at its reticulations and its edges. You'll notice, I don't know if you can see on your screens, especially you can see it on this ridge, how porous it is. I've noticed that the seed seems to be friable and easily eroded, even when fresh. And that was one of the big things that I noticed. I also saw, it's maybe not so clear in this picture, that many of the seeds had a curved edge on one side. So as I said, I had a bit of a workshop. I took part in a workshop yesterday at the Canadian Seed Analyst meeting, and I described this as it was like a couch potato seed. It looks rough. It looks like it hasn't really seen daylight in a while if it was a person. Take that in contrast to the purple toadflax. It's a smaller seed, and I characterized this in contrast to the striped toadflax, and it looks like that these seeds have been to the gym. They've got well-defined ridges, they've got well-defined edges. The edges tend to be thicker. I don't see that same porous erosion kind of a surface that I do on striped. And again, these are smaller, and I do believe that purple toadflax is another species that's used for flowers. Now on to the metallics. Yes, these 2 metallic species do look quite similar, and there is a good reason for that. I hinted earlier, and I'll go over a slide on that. The Dalmatian toadflax, which is by far and large the more commonly encountered of these two species, is a bit of a larger seed. And if you'll notice, the interspaces on the seed are quite small relative to the seed size, in contrast to the broomleaf toadflax, which tends to be a smaller seed. And notice that the interspaces here are quite a bit larger than the fine lacy faces of the Dalmatian toadflax. Both of them do have a metallic wash. I do believe I can see sometimes some differences with the warmer tones in the Dalmatian toadflax and in the cooler tones in the Genistifolia, but there may be overlap. So I'm just going to put that out there as a potential feature for other folks to take a look at and see if that works for them. But those interfaces I found fairly consistent with the variation within the species. So as I said, indeed, there is a reason why one can hardly tell these 2 species apart, because they are closely related. At one time, Linaria dalmatica, Dalmatian toadflax, was a subspecies of Linaria genistifolia. And just in the last decade, and it takes a long time to catch up, believe it or not, it was split into separate species. So now we have 2 separate ones. So we have found here in the lab, trying to source seeds of broomleaf toadflax is difficult, because when we go into sites to request seed or catalogs, if we see Linaria genistifolia, we are aware that it may refer to either species if a subspecies is not given. Because remember, not long in the past, they were both Genistifolia. They were both subspecies of Genistifolia. So a lot of times, just in terms of shortcuts or perhaps not understanding the significance of saying what subspecies you had, very often Linaria genistifolia, without any subspecies, often refers to Dalmatian toadflax. Like I say, maybe in the interest of space, or folks may not have seen it as important, so they just say Linaria genistifolia. So now in catalogs and sites, even though they are separate species, if they list both species, then we know genistifolia is actually genistifolia. If they don't, then look for that subspecies or be aware that you may be getting Dalmatian toadflax. All right.